My name is Craig Frey. I am the commercial vegetable agent here in Southwest Florida. We thank you for joining us on the meeting and those who are joining online. This is more of kind of an update meeting. Some of you know that we've had shifted a little bit to more industry-focused meetings. We've gotten to do that, especially on the tomato side of things with the Florida Tomato Conference, joining with the Florida Tomato Committee and putting that on. This past year, we got to have the Florida Snap Bean Conference kind of for the first time. And more in Belgrade being hosted. There's also some lettuce meeting and the sweet corn meeting that happened there. But so we kind of have those industry-wide meetings, and then we also have these that are a little bit more general in topic and focus. So that's what we get to do today here, and some updates from our faculty members on weed and disease management. To start out, I'm going to talk a little bit about what's happening on the extension side of things. So we've got this new commercial vegetable extension website, and basically the idea is it to be a go-to place for all of the work that we're doing extension, all the information that we're trying to get out to producers. Instead of there being multiple places to go, now it's in one place. And there's so we'll have information about upcoming meetings on there. That's where the recorded meetings are. Our pest and disease hotlines that we send out every 3 weeks are on there. The archives are on there, and we also have kind of this research connection section, which is just an article that we put out every 3 weeks about some research that is applied research that has the potential to benefit you and your production system or your growers if you're a consultant working with them. So that's kind of what's going on that production vegetable production website. There's also information about CEUs, and you can get CEUs from previous classes that we posted on there. There's also pesticide prep tests classes for prepping for pesticide tests. Those are on there as well. So the idea again is to kind of compile all that information for you. So I'm going to talk a little bit more about what's happening, and we're really focusing, what I'm focusing on today is more what's happening here in South Florida between myself, Michelle, who's my assistant, and then Anna Mazaris is the Palm Beach County Vegetable Extension Agent. So we work a whole lot together. Again, we're kind of pulling and trying to build more of a team approach as opposed to everyone doing their own things. So the kind of the highlights of what's been happening in Extension is this collective effort that we've been doing. So some of you are aware of some of these new pest assessments that we've been doing, Asian bean thrips for the past couple of years. This new weevil pest, some people are calling it a carrot weevil because it's similar to that, but it's actually a different species that has been causing some issues. And then a new thrip species that just seemed to rear its head in peppers in the last month or so. So I'm going to tell you a little bit more of what's happening with them. So the Asian bean thrips, this is something that was causing in 2020 was causing about 30% losses in homestead and snap beans. And so basically what we wanted to do is just to get as much information as quickly as possible to help growers avoid losses. We didn't know, we knew it had come up in our area as well that first season. And so we started doing some scouting and surveying assessments, working with the commercial scouts as well as being out there ourselves. To get an idea of when does it show up in our area, what population levels are actually causing damage, what things are working to manage it. And so we end up throwing this together, this very rudimentary website that was kind of built over time as we learned over the last 2 years. At one point we were doing some scouting reports, the weekly scouting reports, sending that out. At the end of through most of last season, the populations actually remained low in this outside of homestead. So we kind of felt like we didn't need to fill up your inboxes this year with all that. If things do spike, we'll obviously keep you all informed and let you know through the hotline. And if it's absolutely necessary, we'll do some of those weekly scouting reports again. But so that's kind of where we've been with that over the last 2 years. We did have the first season, we did have some growers in this area that did have some damage. So there's a potential. We're not really sure why it spiked 2 seasons ago and not last season. So we'll see what happens as we continue to monitor that going forward. This new weevil is a pest of the Apiaceae family. That's the celery, parsley, carrot. Those are the main crops that it seems to be damaging. Anna is the one that has spent the most time working on this and has already done some insecticide trials. This was some very preliminary stuff from last season. There's some more information from this season that she did some on-farm trials, and that was just completed just before the holiday break. So she's compiling that, and then we'll be getting that all out to you to help those growers be able to continue to manage this pest. It basically bores into the stem of the stalk or the crown of the plant and causes plant death that way. It's something that it sounds like it's been around for potentially from the commercial scouts that we worked with for the last 4 or 5 years, but wasn't really ever a major issue until last season when it spiked quite a bit. Have even found that some blacklight monitoring has seemed to be really effective in helping us see when populations are coming into a field. So we're continuing with that. And this new thrips, species thrips, parvospinus, is something that has been in Florida since 2020 and was mostly just a pest on some ornamental crops and in some nurseries. Some of you, most of you hopefully are part of the hotline, pest and disease hotline that we sent out. If not, please let me know. I'd be happy to add you to that. But most of you probably saw that the last 2 versions right before the holidays, there was an outbreak that was causing a lot of damage in peppers in Palm Beach County. So we're continuing to monitor that. It seems like a lot of that was because the field was close to an area where plant debris was being brought, mostly from the coast, these ornamentals. And so because of that, there was this huge refuge, as well as importing of this species from the coast to those pepper fields. So the little that I know so far in this area, talking with a couple of growers and scouts, and when we specifically sampled for this, we haven't found this yet here, but it was causing some severe, it's a foliar feeder, severe stunting of the plant. So again, we'll kind of do it similar to some of these other things. We'll keep monitoring it. We'll working with commercial scouts and ourselves out in the fields, assess where it is and when it's there, and what things might be working for managing it, and try to let you all know as soon as we can mostly through the pest and disease hotline as well as the website. Some other pest ID information, things that are being finalized, that are still being compiled. 2 years, or I think it was 3 years ago, 3 to 4 years ago, there was in tomatoes, there was a pretty high spike in stink bugs. That typically stink bugs is a pest of tomato in northern states, not as much down here. So the Florida Tomato Committee sponsored some surveying of that, and so I've gotten to be a part of that, and my assistant Michelle as well. And one of the things that some of the growers had asked for is a guide to the nymphs. So this is just something that we're working on. Hopefully by the end of the month, we should finish this. But as you can see, the nymphs of some of these species look very different than the adults, and even the different stages of nymphs. And so this is the type of thing that we're putting together on all the different species we're seeing around tomato fields. The thing, unfortunately, fortunately for our growers, the last couple of seasons, the stink bugs hasn't really been much of a problem again. We see a lot of different species around tomato fields, so it's hard to know exactly what is causing the damage. It doesn't seem like there's a primary species. It seems like it's just this whole complex, a little bit of each. But hopefully this will help, getting this in your hands, help us all be able to ID these well and know what is in our field, particularly if we have a year where they become bad again. So another thing a little bit similar is putting together a guide for weed hosts of cucurbit viruses that has some symptoms. This is actually mostly whitefly transmitted viruses at this point. So ID pictures, which most of you are pretty familiar with some of these viruses, but making sure that we understand what weeds are also hosts of these. because often where we see some of the worst damage in some of the melon fields the last 2 years that I've been out here and out and about is where these are all on the ditch bank or in a not natural area. So balsam apple is one of the big ones, amaranth, and some others. So again, this is we're finalizing this, and we'll be communicating this and just getting this information to you all. And if there's other information like this that you guys are really interested in, that we can work on putting together to help you all, things that aren't really out there or hard to find on the internet, some type of ID guide, please let us know so that we can be a part of working on that. Another thing that we're involved heavily in is the soil moisture sensor network. And basically, this is a statewide network. We install these most of them have Syntec sensors, and then we see what we can learn from it. So these are sensors that have soil moisture readings at multiple depths, basically every 4 inches. And it gives you eyes under the ground and helps us better understand water use, water holding capacity, and potential impact on nutrient availability. So for this season, I just have a couple left. Most of them are out in the field, but this spring, if you or one of your growers that you work with are interested in trying a couple out, put some in the field yesterday, putting another in a field tomorrow, but I have about 2 left for the spring season, and be happy to get it out there. So probes are generally around 2,000 each. Cost share options are available, but we have these at UF-IFAS Extension. Between me and other counterparts, I can, if enough of you are interested, I can get probes from other places around the state that aren't being used, and we can get them in your field, so we can try it out and learn together. There's a couple success stories, but I don't really have time for them. Our next vegetable grower meeting that I'll mention again later, we're going to be focused a little bit more on, or exclusively on, irrigation and nutrient management. And so we'll talk a little bit more about this, but some of our seed companies and working with some of our large growers have all had some very significant success in reducing their irrigation and being confident in not stressing out their plants, because they can now see what's happening 4 inches under the ground and 8 inches under the ground. So another thing that we're trying to get more involved in is some industry trials. So those of you who are industry folks, if you're doing trials with growers, we'd like to partner with you in that. We'd like to help on the growers. We like to help them make sure they're understanding things as best as possible and making good decisions from the results that they have from these trials. And then it also is information that we get to share with our network that we have. And so as we see success, this is a fumigant trial, or this picture honestly isn't actually from it, but involved in doing some low rate fumigant trials in beans, seen some success with growers in Central Florida, and doing that with some growers now. So anything you all are doing on farm, we'd love to engage in that. There's some of you are familiar with some of the BMP trials that are happening, the nutrient management trials that UF-IFAS have been funded. So these are all the different projects statewide. There's $8.8 million that the legislature has given through FDACS to UF for nutrient management studies. And so many of you who have been doing this for a long time know that growers or you yourselves have never really been happy with many of the nutrient recommendations that IFAS has. Some of that is because some of the research was done 40, 50 years ago. And obviously yields are a lot different now than they were then. And now we're in a place where we actually have funding to do it. We haven't really had access to funding for a while, but because of things that have changed in the legislature, it's become more of a priority. And those of us in Extension, particularly down here, involved with the team with Sanjay Shukla and Shinsky Agahara. Shinsky's up in Central Florida, but we're working as a team looking at tomatoes, potatoes, and snap beans. Again, I'm not going to go through this too much, but we're going to share some of our results at the next meeting in these crops. But basically, we're seeing that the previous change from Malic 1 to Malic 3 conversion isn't really appropriate for all soils. Tomato stuff is ongoing, but we have seen significant differences in crops irrigated or with phosphorus fertilizer versus those without. So we shared some of that at the tomato meeting for those of you who were there from last year. Again, in another month or so, we'll share some information that also includes some data from this season. And then the potato, from some of this research, they've also had some previous years of research. This year, in end of October, IFAS put out a statement where they were having a provisional phosphorus rate recommendation, and that basically, regardless of your soil p-test, you were able to use the maximum amount, basically saying we have enough evidence that what our current recommendation isn't working, and we don't want to penalize growers. So we're doing this provisional recommendation until we can have enough data to have a rate to be able to give based on soil types and things. So that's where we're going with that. I've been involved, especially more on the snap bean side of it. Tom Obriza is the one who is kind of the head of that. He's in Gainesville now. He used to be the soil scientist down here, but he told me to let you all know if anyone has any questions. He's the one to contact. How crops are prioritized is honestly a little bit more on the political side of things than anything else. So if it's the growers that are the most vocal, they're the ones whose crops we've started on. But obviously we need to, for down here, melons is another crop that we need to add, peppers, some of our other cucurbits, sweet corn is another big one that we need to add to what we're doing. Most likely tomatoes and potatoes will be finished after next season. So then our team will have the capacity to start adding some more crops and going from there. Just a minute left. So there's some other random initiatives where we're trying to to support our growers as best as possible. So Hurricane Ian came through, and what we realized, this will take too long to get to where it was, but basically here in Southwest Florida, these are some of our fawn weather stations, and that are recording wind speed, and where the storm came, there's a huge gap. And we're still trying to understand exactly, here's kind of all the weather stations from NOAA, still trying to understand which ones on the coast might actually be considered official or not. It sounds like if it's at an airport, it's official, or they'll use the fawn stations. The rest of them all just have temperature information, no wind speed information. And so how this has affected our growers is some growers have not been able to get insurance coverage for crops that were damaged by the hurricane, wind damage by the hurricane because of this. So we're trying to figure out, can we get some funding from USDA or the legislature here to be able to put some more in Southwest Florida to have coverage. The policies basically say if you have, has to be sustained hurricane force winds in your county or the county adjacent to you. So which pretty much makes it for most of our storms really mostly on the coast. But again, we don't quite know because we didn't have sensors in those areas this year. So that's just one thing that we're trying to do. Another thing is next week I'm heading up to Gainesville to I've previously had some conversations with the Dean of Extension and the Vice President, Scott Engel of UF-IFAS. But we're looking at doing kind of more of an official proposal. We've had some stakeholders meetings about creating a new position that their whole focus is agriculture awareness, educating folks on the coast in South Florida about agriculture, how things that we're doing to be sustainable, things like that. So all of us in Extension do this in some ways. Many of you all might, educate people here and there in different conversations. I think it'd be really valuable to have someone that this is 100% their job, getting in different media as well as social media avenues, telling grower stories with fact-based information, and then also again kind of helping all of us have better information to share as we do that. So the last thing real quick is there's a group here in Southwest Florida it's kind of like Fresh from Florida, only it's Southwest Florida Fresh. It's really geared a little bit more towards the small growers, but just wanted to let you know that if someone's interested, some of the larger growers also do some direct to consumer marketing or focusing down here, be happy to connect you with this group that is Southwest Florida Fresh. So that's kind of a whirlwind of what's happening on the extension side of things. And our next speaker is PM Roberts. So let's go ahead and switch this up.
All right, good morning. So today I'm going to be talking about some of the things that have been happening in the cucurbit crops. Primarily talking about watermelon, but this is also applicable to squash and some of the other cucurbits that we grow down here. And specifically, I'm going to be talking about our virus complex and a survey that was done last spring. I'm going to touch briefly on the powdery mildew and some fungicide screening, and then talk again briefly within the time of a couple of bacterial and a fungal disease that seems to be emerging on watermelon. So we know whitefly transmitted viruses and the whitefly vector are major problem disease here in watermelon production, and it affects squash and other cucurbits. I'm not going to be talking about management today. I will refer you to this EDIS publication that was just updated this last year that has all of this information in it. But basically, when we're dealing with managing these viruses and whiteflies, we're looking at sanitation, we're looking at cultural controls, we're looking at, of course, insecticides, and using a whole range of IPM to try to manage the whiteflies and viruses. So the virus complex, as we know it right now, there are 7 viruses that are occurring in watermelon, and most of these are also in squash and zucchini. Our old friends, or old enemies, nemesis, squash vein yellowing virus. This is a whitefly transmitted virus, showed up about 2003, devastating. It was killing watermelons, adversely affecting the quality of the fruit. Cucurbit yellow stunting disorder virus. This was also found in Florida in the around 2005, 2006. Cucurbit leaf crumple virus. Again, these are both whitefly transmitted virus. We've been dealing with them for about 15 years. And then the potavirus complex. The predominant one in South Florida has been the papaya ring spot virus, but there are other ones, and these are the aphid transmitted viruses. Now this lower tier is what I'm calling the new viruses. So beginning around fall of 2017, we were getting some feedback that the growers were seeing something different in their crops. And we had been working with Matthews Perrette on trying to validate some rapid diagnostics for the viruses. And so we were getting a lot of samples in of cucurbits. And we would receive samples that look like they should be viruses, but when we detected it for the viruses that I just talked about, the squash vein and cucurbit crumple and all of those, they were coming up negative. And I happened to be talking to a colleague in Texas around 2021, and he said that they had just described some new viruses in Texas. So we collaborated and sent some samples out to him, and we did identify 3 new viruses to South Florida, 2 of them new to Florida. So the new viruses are watermelon crinkle leaf associated virus 1, watermelon crinkle leaf associated virus 2, and then cucurbit chlorotics yellow viruses. So this was the first report of them occurring in watermelon in Florida. What's unique is that these WCAV-1 and 2 are a completely new virus type. They're different from the ones that we've been dealing with, and actually this is they're pretty new worldwide, occurring in plants. So for these 2 viruses specifically, they were first reported out of China in 2017. My Texas colleagues described them in 2021. And as soon as Florida found it, there were reports out of Brazil, North Florida, Georgia. So the geography of these are increasing. They do cause symptoms that are similar to known viruses, which is why we were, kind of picking them up. But it took me a while before I really could go out in the field and say, okay, yes, this is associated with these 2 viruses, or one or the other. Now for this group of viruses, again, these are pretty recently identified. So for the plants, there's not been any vector yet described for them. But for other viruses in this group that go to human, animals, or other plants, not cucurbits, they're vectored by arthropods such as mosquitoes, flies, ticks, plant hoppers, and thrips. It doesn't rule out whitefly as a potential vector. That's one of the things we've been looking at. But right now, there's no insect vector associated with it. We're also looking at modes of transmission by seed and mechanical transmission. And one of the problems that I'll probably mention quite a few times in dealing with these new viruses is because of all the other viruses that we have out there, it's really hard to try to get at exactly what these new viruses are doing. So the other virus, the Cucurbit chlorotic yellow viruses, this again was detected in pretty much South Florida and North Florida at the same time. This is not a new virus. It's been in the Middle East and North Africa. It was in California, Arizona, and then it made its way to Florida and Georgia. This is the crinivirus. It's in the same family as the Cucurbit yellow stunting disorder virus that, again, we've had here since about 2006. Very similar symptoms. Again, it affects fruit qualities. It's whitefly transmitted, and it does go to cantaloupe, watermelon, and squash. So getting back to the new viruses and the overall complex, I mentioned one of the first things that I needed to do was kind of be able to distinguish visually the symptoms and then follow up with the testing in the lab. We've been looking at a survey, not only from going out in the growers' fields, but also from what's been collected in the sample in the clinic, trying to determine, the incidence, how frequent they occur, and the geographic range of them. The transmission is a big issue that we're trying to look at. And then also when these two viruses were first reported, it was only in watermelons. So we wanted to look at squash and other cucurbits and also weeds. And then finally, the so what? So we have these new viruses. Are they doing anything? Is there any kind of economic impact? Are they, affecting the yield? So getting to the symptomology, I'm getting pretty confident now when I go out in the field. If I see symptoms that look like this, where they're crinkling and kind of, what do I want to say, crinkled, and they kind of have a predominant white vein in the center, these would be highly suspect, and I would be pretty confident that they were going to test positive for at least the WCLV1. Just a side note, the WCLV-1 is a little bit more predominant. We do pick up the WCLV2 occasionally, but they have pretty much the same symptoms as far as we can determine. So last year, in the spring of 2020, I put out some plots in various watermelon fields in Collier, Hendry, DeSoto, Charlotte County, and kind of marked off a section of the field. And then I went back at 2 weeks intervals to try to look at the progression of the viruses in these crops. I'm also going to include samples that we received in the plant diagnostic clinic at this time. And also at the same time, we were pulling samples from squash, zucchini, and weeds to try to look at the host range. So for the crops that were planted beginning in December and then throughout the spring, started surveying in February when we pulled the first sample, and at that time I detected only WCLV-1 and -2 in the very early stages of the crop. But 2 weeks later, we were picking up squash vein yellowing virus. And then as the season progressed, we detected all of the viruses. So by the end of May, we were pretty much detecting all of the 7 viruses. So I want to show this first. This is one of the last sample dates that I had at one of those plots that I'd been monitoring over the season. And the WCLV-1 was in almost 90% of the samples. It was into, or 12 out of the 13 samples that we pulled from those plots. The next ones that were highest in incidence was the CYSTV and the WCLV2, the squash vein yellowing virus, podivirus, And that one was not detected. So that was kind of the distribution in the field at the end of the season at one site. Now, I wanted to show that because that follows the trend throughout the entire sampling season. And I don't think that, you know, there might be some interference with, you know, some of these viruses that showed up later in detection methods, you know, the level of sensitivity. But overall, the same trends held for all of the samples, almost 200 samples pulled out throughout the season. And again, this was the predominant one that we found in the samples, followed by these two and those. So there's no question that everything's out there. So we've got a mixture of 7 viruses in the watermelons. And then more recently, I just want to kind of touch on this in passing because we're trying to get at the mode of transmission. And in the fall of this year, we brought some samples, some transplants into the clinic. And when we tested them, they were positive for WCLV-1. And so we went back and were able to obtain some of the seeds of these transplants. We planted them, they grew, we tested them, and yes, they were positive. This was in a controlled environment in our confinement, so there weren't any whiteflies, there weren't any insects. And so from there, we decided to try to do some greenhouse isolating them to see if we could get at the effects of the WCLV-1 only on the watermelons. And we just pulled these trial down right before Christmas and after Christmas break. So I don't really have any data to show you other than I think we probably need to work a little bit on growing our watermelons in the greenhouse. We've got them tied to vines here and we're trying to get fruit. And so this is going to be repeated and hopefully I'll be able to get at some of those questions, particularly the effects that it has individually on the watermelons before, hopefully. So finally, just to kind of wrap this part of it up, so we're finding the new viruses, the WCLV-1 and 2, and very high incidence in the field. We're detecting them earliest, as the information from the transplant detection seeds would suggest. It's pretty much throughout South Florida. It's been reported in Georgia, so geographically it is increasing. Again, just want to repeat, because of doing anything in the field is confounded because of the whole mixed virus complex. It's really going to be difficult to do anything in the field to try to get at exactly what these new viruses are contributing to the overall virus situation. Since I gave this talk last August, parts of it at the expo, squash and zucchini have now tested positive for these 2 viruses. I've been pulling random weeds associated with the watermelon fields and squash fields, and so far we've not had any weeds that have tested positive with PCR for these viruses. We're looking at seed and mechanical transmission, and of course the bottom line is, so what? What are these viruses doing? And hopefully we'll be able to continue to get some information on that. Now, in terms of the overall viruses, again, there's no recommendations right now in making any changes to the management practices. I'm continuing this work, and again, I would just like to refer you that in management of the whitefly and these viruses in this recently updated publication that's available on EDIS. So, powdery, okay, going back to this emerging diseases theme. So in watermelon, powdery mildew seems to be getting not only more frequent, but more severe. In North Florida, it's a major problem. In South Florida, we're starting to see it pretty regularly in the crops. And powdery mildew, of course, as the name implies, it causes this kind of powdery film on the leaves, and it can also go to the fruit, which affects the marketability. So some of the issues with the powdery mildew, we're not really sure why it seems to be becoming more aggressive and more widespread. But in other regions of the US, they have reported that the population is developing fungicide resistance to many of the FRAC groups, representing 1 3, 11, and 7. So we started looking at that with the population down here and trying to do some screening to see if the fungicides were still effective on the population of powdery that we had on squash and watermelon. So this work was actually initiated right before COVID, and then we had the research pause, and we're still trying to get it back up to speed. But we did do some initial screening using these different fungicides representing the different FRAC codes here. Of course, this is the mode of action and the product names. And we did the screening at the field rate. We did it both on disc and plates in the lab, and also we did it on seedlings in the greenhouse. And just to show some of the results, and we have a lot more data than that, but this is kind of the take-home message that was consistent across all of the different trials. If we look at disease control, of course, with the water, there was 0% disease control. The Topsin also across the trials didn't do anything. The Torino here was, I'm sorry, the Rime. We had kind of variable results with that, and some of the repeated trials that worked very well, and other ones we didn't get very good control. So that was kind of an intermittent product. Quintec, Raleigh, and Torino, they all seem to perform pretty consistently well in terms of management in our trials. So we're going to be continuing this work. We'd also, of course, like to do it in the field. The situation in North Florida is that Matthew spread has a little bit more consistent disease pressure on watermelon. I have consistent disease pressure on squash. So we're kind of trying to combine our efforts to continue looking at this and include it to some of the fungicides that were more recently reported that the population was developing resistance to. Okay, so last part of the talk. We all know about downy mildew, gummy mildew. Those are pretty, chronic, and our crops get them year after year. But more recently, we've been picking up some other things which have not been quite as common. Now the Pseudomonas, we've had sporadic outbreaks of this throughout the year. But again, it seems to be increasing, particularly when we've got the cool, wet weather. Beginning about 3 or 4 years ago, we started to pick up a bacterial spot caused by Xanthomonas a little bit more consistently. And then anthracnose is also, that's not a new disease, but it's something that we haven't typically found as consistently and as widespread as what we've been picking it up recently. So I wanted to put these diseases on everybody's radar. And again, if you look at the symptoms, sometimes there's not a lot of difference between the symptoms. And we've also had the anthracnose. Gummy stem will also get on the fruit, but we've also had anthracnose on the watermelon fruit. So again, just for the sake of telling you it's out there, getting on your radar, picking it up sporadically, but picking it up more frequently, both the Pseudomonas and the Xanthomonas. The anthracnose seems to be showing up pretty much season after season. This is caused by the fungus Calototricum. Historically, it's been caused by one type of Calototricum, but again, recently there's been reports of new species occurring of the Calototricum on cucurbits. And one of the things that I'd like to look at is which Calototricum we have, because that might have implications as to the sensitivity to different fungicides. The reports are that this anthracnose is very difficult to control in the crops. And I'll get back to that. So I also wanted to present some research that Matthews Pret did on managing the watermelon bacterial spot. This is trials that he did up in Quincy. And basically, as you know, for bacteria, we've got very limited materials available to us. Basically copper, some mancozeb, and then the SAR compounds. In this case, it was Actigard. And some results of trials that he did. in the spring of 2020 and then the spring of 2021, looking at co-side with the mancozeb material, either alone or in combination. It did give about the level of control that you would expect on these compounds, you know, for managing bacteria. We never get as the level of control for a bacterial disease that we do for a fungal disease, but if we can reduce it by about 50%, that's a pretty good ballpark. And then in the second trial where he did some different treatments, the co-side alone, the mancozeb combination, and then with the Actigard, he did again get pretty good disease control compared to not doing anything. All right. Finally, you know, identification of the disease and the pathogen out there is pretty critical for making your choices for management. Both Matthews and Gary Valad have been working more on characterizing the population of Pseudomonas by Perret and the population of Xanthomonas by Valad. And both of them are finding that it's not caused by a single species within that it's a complex. The anthracnose, I'm just beginning to look at this and hopefully I'll get a program started on it soon. But one of the reasons why we may be having so much trouble controlling in the field is because the strobilurins have historically been very good at controlling anthracnose, but there has been reports that the pathogen population has been developing resistance in other parts of the cucurbit production region throughout the US. So I'd like to acknowledge my team. Dr. Catherine Hendricks in my program has been responsible for all of the virus work, running the PCR. She's done a ton of work on this and we've been supported by other members of the team. I've been collaborating again with North Florida and my colleagues at Texas A&M. Funding sources, acknowledge them. Thank you. 20 minutes, right? Yay.
Any questions for Pam as I switch out the next presentation?
Yes. No, that's fruit blotch is caused by a totally different bacterium. It's caused bacteria. It's Ampatzidis. And I didn't mention it. Normally when we see that, it's usually picked up at the transplant house. It has distinct symptoms. Usually it's on the cotyledons. What I'm talking about with the Xanthomonas and Pseudomonas are more field outbreaks. So no, for the Pseudomonas, again, it used to be Pseudomonas lacrimens, the angular leaf spot, and what it is now is a complex of Pseudomonas syringae. So it's not a clonal population within the syringae. Most of what we've seen is on the foliage, but it can really tear through a field when the conditions are right. It can take the vine, yeah. Yeah. Okay. All right.
Great, thank you, Pam. And we'll have a 10-minute break after the next presentation where you can also ask some questions and use the restroom before we have our final presentations. And as you heard from Pam's stuff, all of the work that we're doing, I meant to mention it. When I showed our pictures in extension, I listed all the research and education centers because all the stuff that we do is in collaboration with all of the research and education centers all around the state. And as you heard with Pam and the work that she's doing in North Florida as well, we wouldn't be able to do any of the surveying stuff that we're doing in insecticide trials without working together as a team. Obviously, on the disease side, it's a little bit more complex, especially with these mixed infections that she's been working on. But thank you, Pam, for that. All right, next we have Yiannis Ampatzidis, and he's going to be talking about AI-enhanced pest and disease management. Thanks, Yiannis.
Good morning. It's really nice to see some new faces. Today, I will focus more on the disease detection and classification using AI, artificial intelligence-based tools and techniques. But before that, let me start a little bit with these new systems, new technologies, and we have some issues here with the videos. So I'm not sure if you're familiar with this system. This is for precision weed management. As you see from the left to your right, we have a system developed by Two River Technology, now owned by John Deere. Its idea is speed, spray using machine vision, detect the weed, distinguish from the crop, and spray on the weed. Then the carbon robotics, they use laser, but they also use machine vision and AI for the detection. And the third one is what we're developing here, my team. And what also we do, this is just a demonstration, what you see here, is like we can also classify different type of weeds. And we can even spray only on a specific weed if needed. And we have a grant to develop, to further develop this technology. And we, in collaboration with the Carnegie Mellon University, we develop also a robotic system. Again, this is a prototype. This is for the vegetable production. We have some videos here to see how this is the latest version of the system. It's adjustable. I hope it will start. It can fit in different spacing. So it adjusts its width and the height, and we are going to attach there the sprayer that we're developed here. And again, we utilize AI to see and spray. This is the idea, and we can use it for on the bed, treatments on the bed, but also within the rows. And then you see here in the right video how we can adjust the width of this machine. And again, this is just a prototype. If we try then to develop a system for our specific production system, it might be totally different. But again, this is an idea that we are trying and to see if it's effective and economical feasible. So disease detection and disease classification. And keep in your mind this word classification. So the first one is the symptom-based. I'm sure some of you have seen tools like that. They're commercially available. So I just put an example here that I'm working with my team. This is not published yet, but as you can see here at the top, we have some, I think 6 diseases and a healthy leaf. I will try to focus today on some of the challenges and limitation of these technologies. And sometimes it's very difficult to develop accurate detection system. And this is again, detection and classification, because we detect that there's a disease and also we try to classify it. So as you can see here, there are going to be a lot of, in the future, web applications or mobile apps that you can take a picture of a leaf with the symptoms and it will tell you or try to detect and classify the issue. It might be a disease, it might be a virus, it might be a disorder. So that's why it's challenging. These systems are like machine learning, AI-based technologies and require a lot of images to be trained on. So in order to develop this system, we need a lot of images. And this is number one challenge. And this is number one mistake that I see. A lot of times we go to the field, we see some symptoms, we take a picture, maybe we have an expert with us and we classify it without really even doing some kind of lab analysis or chemical analysis. That is sometimes very, very problematic, because let's say that I want to classify bacteria spot or something like that, but I find something that it's really target spot. You know, diseases, they might have similar symptoms, especially at the early disease development stages. So this is the #1 problem that I see. Companies, they go out there, they collect pictures, and they don't really know for sure that these symptoms caused because of a specific disease or vector or something like that. So again, this is very challenging. It takes a lot of time, a lot of effort. But again, as Craig said, we are more than happy even to work with you if you want to develop a technology like that. We have the experience. We're already developing systems like that, but we don't have anything that is like a commercial system right now. We are not there yet. So we are doing more processing. Everything is automated. So what I describe here, there are some techniques that we use. It's not just we use machine learning or classifiers. I'm not sure you're familiar with that. I'm not going in details today. But what we try to do, for example, first of all, remove the background. That's sometimes really helpful. Then another technique that we try to use is maybe focus only on the symptoms, remove the healthy tissue from the picture. And then as you see here in the table, you see all the classifiers. I'm going to use this word, classifier. This is AI-based techniques. And we have different ways to analyze the image. We can focus on the color features, we can focus on the texture features, or we can focus on the shape feature all together. And here you see the overall accuracy. The higher, the better, of course. If it's close to 1, that means 100% detection accuracy. And at the end, the #8 is like a new technique that combines all these classifiers, not equally, but based on their prediction accuracy and known prediction accuracy. So we try to develop, again, different techniques. We call these steps pre-processing, and then run the classifiers in order to see how accurate we can be. And it can be very accurate, especially if we have a good quality. The quality of the data set is a key, and of course, the quantity. But again, this is a symptom-based detection. So we need to have the symptoms to detect a disease or a disorder. And again, we need examples also from, let's say, nutrient deficiencies, so we can compare with that. It might be some symptoms because of some nutrient deficiency or something else, so a different disorder. So we need to develop to have a very good data set. So again, if you think that something like that will be useful for you, we are more than happy to collaborate with you. We try to find some funding. It wasn't easy to develop something like that, but we are going to continue for different crops too. Now, early detection. This is the next step. Early disease development detection. So how we can detect a disease at the early stage. Sometimes we call it even the asymptomatic stage. This is the stage that we know that the crop is infected, but we don't see visually symptoms. And let me a little bit explain that. And before going there, let me present a little bit these technologies, hyperspectral remote sensing, hyperspectral cameras, and multispectral cameras, and what is the difference. I'm not sure if you're familiar with that, but here what we see, let me start with the RGB first. The RGB, they're the normal cameras that we use. We have in cell phones, all the cameras, we call them RGB, they're actually red, green, and blue. So what happens here is like every object absorbs part of the solar radiation, the solar light, or the ambient, any light, and reflects part of it. So what we see, the human eye see the reflectance. But what we can really see, the eye is a very nice tool, but it's very, very limited. We can see a very small area of the spectrum. So this is exactly what we see here. Now going to the hyperspectral, with the hyperspectral cameras, they have the benefit, or what they provide is like they can take data in a very, in a few nanometers. So we can create this line, which we can also call signature. That means we collect a lot of data. The multispectra now, we have these histograms. For example, in the blue or red area, might have like 2 measurements. And this is a problem of them because we don't have standards in the industry right now. We have a lot of multispectral cameras that might focus, let's say this is the red area. If I focus in this wavelength, again, the x-axis is the wavelength, the y-axis is the reflectance, the intensity of the light. If I take a measurement here, it might be different if I take a measurement from this point. And this is very common in the industry. We have companies that develop multispectral cameras that they focus in different wavelengths. So we need to take that into account when we develop any kind of remote sensing system. So again, what we try to do here, we try to create this kind of signature, especially with the hyperspectral cameras, which is the expensive ones. That's why we need to develop low-cost sensing system, which is more like the multispectral type of camera. So as you see these lines, or again, signatures, we call it, the green one, for example, is for a healthy plant, as an example here. The red is a stressed one, then we have a nitrogen deficiency. It's like a fingerprint. So this is a signature of a plant, which is very, very important, because if we know these signatures in different growth stages, we can identify differences. Of course, it's not that simple. There's a lot of overlap. And then what we can do in a map, we can say that, okay, this is healthy vegetation, a healthy crop, this is stressed. We can even detect weeds or find nutrient deficiencies or diseases. But again, let's focus on the limitations or challenges. When we collect the data using drones or satellites, is very critical. If it's sunny, if it's cloudy, the time during the day, all this information that are very critical. We need to calibrate the sensor every time we collect data. So, and plus after that, we need to clean the data from the noise. And this is what we try to do. Also, we try to understand what happens in different wavelengths. Again, here you see the wavelengths. Here is the reflectance in the y-axis. So in some of these bands or areas, we know now that they can represent, for example, the chlorophyll absorption, or we can find other pigments. Based on that, we can identify issues, stresses. Without going a lot of details now, let me give you an example of this signature of a healthy tomato crop. So what you see here, again, wavelengths in this axis. This is samples, so 70 or 90, I don't know, I don't remember how many we have here. And again, we call it the normalized intensity or reflectance. It's normalized because we had to clean this data to pre-process the data. So we have this kind of lines. And again, as you see, there are similar, but there are differences, right? Okay, let's see this slide now. At the left, we have a healthy tomato plant. Right, it's one infected with black spot disease. Do you guys see differences? Yes, maybe, no. It's not easy. So if I pick only one of these signature and I put it here, it might look okay. Yeah, it can fit. So that's why we need to use other techniques, and we need to use AI to help us differentiate and classify. And this is what we are doing. I just put here some techniques that we use without really describing. This is just a very simple technique. But again, doing that, we can identify peaks or we can identify differences. So we see this data as signals, and we use signal processing techniques. And then after that, we can use, or with them, we can use at the same time classifiers in order to distinguish and classify the diseases. And again, this one example. So what we have here in this figure, we see healthy tomato crop, tomato infected with bacteria spot and target spot. And again, we work closely with PAM. She's helping us to do the ground truthing, which is very, very important. or sometimes we inoculate the plant. So we need to know for sure if it's infected only with one disease or multiple diseases. Of course, in the field, it's more complicated. But again, things like that, they can give you a lot of information, and especially when we do it in the early stage. Here I put another app that we developed back in 2016-17 when I was in California, but this is symptom-based, just to see the difference between the early detection. That's why I put it here. So, and again, this is some examples of 4 different diseases on tomatoes, and this is a squash, powder mildew. But here, what we see, we try to follow the disease development stages and see, can we detect the disease at the early stage? Even maybe they are symptomatic. They are not visual symptoms. There are changes to the plant if it's infected, but we might not see them. But these cameras might pick them up, and this is what we try to do here. So let me explain a little bit how we work on these trials. We have actually 2 experiments. One is we collect leaves. This squash can be tomato leaf, it can be any kind of leaf, and we take it in the field, and then we collect the spectral signatures, or we fly with the same camera in the field, and we try to see if we can detect in the field. So 2 different experiments. So again, we use the same camera, and if you, I mean, I go a little bit fast, but if you have more questions, I'm available today after the lunch to discuss more about it. So we collect data in the lab, which is more accurate now because we control the light, we control the conditions, and then in the field, which is more challenging because in the field, we might have some early symptoms with late symptoms on the same plant or the same row, but at least we can detect hotspots, and this is the important part. So this is a lab analysis. In the lab, we can have more stages of the disease development. And this is, for example, one, the red one is the asymptomatic. The green one is always the healthy. The red one is asymptomatic, so there are no visual symptoms. And here you see how we can try to distinguish between healthy and the asymptomatic, and the accuracy was more than 80%, which is pretty good. And then, of course, when we have late symptoms, the accuracy is almost 100%. In the field, though, we have less classes, because as I said before, it might be asymptomatic with early, we combine that together. But again, this is the early stage, and still we are able to detect with more than 80% of accuracy. And having more data, having more trials, and we try also to find some funding for that, it can really help us to detect that. And again, as I said, here you see the signatures, and here is a different technique, let me call it technique, that we try to use so we can distinguish between these stages, or later it might be between different diseases. So it's not only we use the signature, we do much more than that. And there are papers if you want to see exactly what we are doing in all of these trials. So I put this technology here. This is a sprayer, drone sprayer from Kiwi is the name of the company. I'm not sure if they are still around, but there are other technologies, like other companies like that, other technologies like that. This can be something that we're going to see in the future also. As you can see, it's not really flying, it's more like hovering above the field. But if we detect hotspot, you might need just to spray on the hotspots, and you can use a technology like that. And very quickly, I know that I don't have a lot of time. We developed this cloud-based, AI-based tool. We use it mainly for citrus. What you see here, again, it's a citrus field. You have some analytics of every block. When you click the block, you are going inside the block, so you know how many trees you have, how many gaps you have, information about every tree, like canopy size, volume, height. You can zoom in. It's very interactive. We want to develop something similar for vegetables. And also, we developed this fertility map. So you see different zones here. And then you see all the colors here, the zones represent deficient to high or optimum. So we can develop something like that. This is again using multispectral cameras and drones. And let me go to the next one. This is a smart sprayer we developed for tree crops. We are working on vegetables again to do the same. It can detect also fruit. Here, for example, it detects a gap. It will turn it off the right side. It will not spray there. It detects the height, and it can change and detect the leaf density. Here we call it at risk because there are less leaves, so we can even spray less there. So this now we're working to combine the fertility maps with this system, so or even with a spreader, fertilizer spreader, so we can apply based on the need. And this is some maps that we developed again. This is a spray map, a fruit heat map. And I will stop here. I have to make two announcements and I'm done. One is like, and I forgot to add the slide, is like we organize a conference, AI in agriculture, IFAS with a lot of other universities. And this is on April 17 to 19 in Orlando. I will be more than happy to have all of you there if you want to join. Can you please forward the email I sent you? We are also looking for sponsors. You can have a table there. Maybe you can present your company. Okay, thank you. And the other one, we just got a grant to develop to use these tools to identify damages after extreme weather events. So like after hurricanes. And for tree crops, it might be a little bit easier because you can find fallen trees, you can see fallen trees, and we can develop the tools, the automated tools to detect it right away. And we try to see what we can do also with vegetables. We can find flooding areas and maybe some damages to the crops. Thank you so much.
Thank you, Craig. Thank you for all for coming. Now let's talk about some weeds. So I'm Ramadas Kanissery. I'm the weed scientist here at the station. So I'm the person who kills plants for a living, actually the bad ones. So this talk, I'm gonna provide you some information on utilizing cultural methods or cultural practices, especially for the fallow month or fallow farm management, with an emphasis of enhancing weed suppression and productivity. So before going deep into the talk, I'd like to acknowledge my co-author here, Diderot Saint-Ilma, who is a graduate student who works with us. And most of the data and information I'm presenting today comes from his master's research project. So in South Florida, when we think about the vegetable production, typically it's been done in 2 seasons, spring as well as fall production seasons. The summer months in between these 2 seasons are kept fallow, meaning there will not be any crop for most of that time. So this is an example of a fallow field in a commercial vegetable farm. So it's something that's been overlooked, but we have to make sure there is a good management practice in place during these fallow months, although there is no crop, because that can go a long way, especially it's going to help us reap a lot of benefits into the subsequent crop season, which I'm going to talk about. So I want to invite your attention to these data sets that we have put together from a Florida Automated Weather Network. This is a rainfall data for about 10 years in Southwest Florida. So as you can see, most of the annual rainfall we are receiving in this region is kind of concentrated towards our summer months or fallow months in terms of in the vegetable production. So it is not unusual to see farms like this in the summer months or fallow months, which has been waterlogged or flooded in this area. So what does it mean to vegetable production? So there are several parameters I want to bring to the table, but I want to focus on 2 things here. This heavy bouts of rainfall and moisture is going to leach away a lot of nutrients, example nitrates or phosphorus from your soil during the fallow month. And this moisture is going to instigate or stimulate a lot of weed growth as well, which is very important. Again, these are there are other factors that's going to change in the soil and in the system as well, but these are the 2 components I want to focus. So this is a picture of nutsedge or nutgrass emergence in a fallow farm after a heavy rainfall. What does this mean to us? Because these weeds, when they grow in the fallow time, if you let them flower and seed, they're going to add a lot of seeds to that seed bank soil. It's going to, have replenish that seed bank with weed seeds, which is going to be a ticking time bomb for your next season, your fall season or the spring season tomato crop, right? So we have several tools in our toolbox for fallow weed management or fallow field management. Most of us, we know when you have weeds, you're going to spray with herbicides. There are some glyphosate programs, Roundup programs for fallow field management. But there are some other tools as well, which I'm going to talk about today, which are cultural methods or cultural practices. Not only help suppress the weeds, but also help us get a lot of additional benefits as well. For example, cover crops. So here is a picture of a sun hemp as a cover crop in a summer fallow field. So cover cropping is not new to crop production. It's been used in several crops, production system, agronomic as well as horticultural, with crop, like in corn. I come from Illinois. I did my PhD in Illinois, and there is a practice of using cover crops in the row middles in corn, and also even smaller levels such as kitchen gardens or vegetable beds. So cover cropping is being used by the growers in several systems, but with crop and without crop, and I'm talking about the cover cropping without crop in vegetable summer fallow, right? So I want to circle back to that nutrient loss issues, which can be a big problem in the crop production season, especially in our soil with sandy soil, right? So cover crop can actually retain. It's a great locking mechanism for this nutrients. So when you have a cover crop, it's going to assimilate all the nutrients into its biomass, and by the time we incorporate it back to the soil, it's going to release it back. So it's a temporary locking mechanism for the nutrient, but it helps in nutrient retention. So we have been doing several projects with cover crops to look at various parameters, including weed suppression. In tree crops such as citrus, which is an emerging practice now in Florida, citrus. So I don't want to show a lot of data. We had 3 years of data, but I just want to show you one picture here to show that how cover crops can suppress weed growth. So in the citrus row middle on the left, we can see our cover crops. It's just cover crops, nothing else. We use daikon radish here. Same area, not just in row middle, no cover crop, but you can see all sort of nasty weeds there, guinea grass, black nightshade, geranium, which can be problematic weeds in vegetables as well. So my point here is cover crops can help in several ways, including weed suppression, if you have it designed or planted in the right way. Another practice I want to talk about today during the fallow field management is compost application. We've been using that. I know there are growers use compost, sometime in fallow, sometime as a part of the land preparation and things like that. It's going to add a lot of organic matter and other nutrition into the soil. Generally, it builds soil quality, and it's very necessary in our region where we deal with this, basically 95% sand, less than 1% organic matter. We are dealing with beach sand here. So, things like this really helps. Practices like this really help. But the crux of our project was to see, why don't we integrate these two practices? Why don't we integrate cover crop with composting in the fallow and see if we can get additional benefits or added benefits in the crop production season? For example, the hypothesis is, okay, the compost is going to boost the cover crop biomass or boost the cover crop growth, and whether we can translate that boost in the cover crop into an added or enhanced benefits into the subsequent tomato production season. So to test this, we had a 2-year study in our station. We had cover crop only treatments. Then we mixed cover crop with compost, like an integration treatment. Then we had compost only and control, where we did not do anything. So in the cover crop and compost, what we did was we let the cover crop germinate. And after 2 weeks, we applied the compost so that it will not be lost in the soil. All the good things will be sucked up by the cover crops, right? So this is a summer or fallow month treatments, okay? So we utilized sun hemp, a mixture of sun hemp and sunflower, a grower, standard use rate. We used a leguminous and non-leguminous mixture because in a preliminary experiment, we found that we have to mix cover crops to get the best out of it rather than using a single species of cover crop. And compost also we used almost similar to a grower standard rate, 6 tons per acre. Then after that, we terminated the cover crop, and in the same plots, we had tomato crop with standard grower production practices, insecticides, fungicides, but we did not use any herbicide program in the row middle so that we can catch that data for weed suppression. So it's going to be really weedy, but still we want to know which worked the best way. So right now, I'm gonna jump a little bit, switch my gears into the some of the interesting results here. So I hope you got the setup of the experiments. It was a 2-year experiment, 2021 and 2022. So as we thought, the presence of compost actually boosted the cover crop in 2 seasons, in 2021 and 2022. Let me set this graph for you. It's a biomass, fresh weight biomass of the cover crop. And we had 2 years, and you can see the bars. There is a cover crop only, as well as cover crop plus compost. Yes, compost worked with cover crop. It improved its biomass, as you can see from this graph. Significantly increased it. Seeing is believing, so I'm going to show you the plot pictures. You have on the left cover crop only, on the right cover crop and compost, and you can see the improvement or the biomass boosting in the cover crop, right? So as I mentioned, then we terminated it by 2 months. One thing I have to bring to the table, we need to make sure the cover crop, if you're using in your farm, you have to terminate it before it goes to flower and seeding, because I have seen introducing cover crop into the farm and it can come back to you as a weed if you do not terminate at the right time. So right time termination is very important when you have a cover crop in a fallow field. So we terminated it and we had tomatoes planted in the same plots. Now, one of the interesting or important parameters we were looking is weed suppression. How we can get a good weed suppression with the compost, with the cover cropping. As I mentioned, cover crops, they have excellent qualities. They will compete with the resources for weeds, and we stop the weeds from germinating. What happens if weeds are not germinating? There will not be any seed production. There will not be any seed addition to the seed bank. So, it's going to suppress, the hypothesis is it's going to suppress the weed in the upcoming season. So this is a piece of data I want to show you. We have dry weight of weeds in the row middles from different treatments. And in 2021, the first trial you can see here, we had 2 seasons. We looked at different time points, and I'm showing the early season weed pressure and the late season weed pressure. You can see in the control, there's an increase in a jump into the late season heavy weed pressure. But it was not shown in other, observed in the other treatments such as cover crop based treatments plus compost as well. That means, yes, there was less weed pressure in that area. There were less weed seed bank in that plots which received cover crop and compost treatments. Then in the next year, the trend was kind of similar, but a little different here. Here, the jump was observed into the compost only treatment, which makes sense because you're adding compost in the fallow. That means you're boosting the soil. So the weeds are also getting boosted. So this is a take-home message that we learned from this lesson, from the trial. If you're using only compost, highly likely you're going to get a more weed pressure in the next subsequent season. But when you combine it with cover crop or use cover crops into the system, you can get the best benefit out of that. So that's that. And the weed pressure in this study, we had very tough weeds. As you all know, goosegrass, crabgrasses, sedges, very difficult to manage ones. And pursleys and common ragweed, hadeotis, all nasty weeds and tough weeds that we see in vegetable production systems. So this was kind of controlled by at least in our observation with this integration of compost as well as cover crop. So again, this is a picture from the trial from the 2021 study. On the left, we have cover crop and compost plot. You can see the row middle clean relatively compared to the control with a lot of goosegrass and the other grasses coming up there. And in the second year, as I mentioned, cover crops plus compost, actually we have less weed pressure. And on the right, the compost only treatment boosted a lot of nut sedge. You can see a lot of nut sedges there. So, but one thing again, we have to keep in mind, we never used any herbicides in the row middles, which means it's not bad. The system we used in the fallow kind of helped, right? So now another important parameter I want to bring is the retention of soil nutrients. We talked about that in the introduction, whether our approach helped retain some soil nutrients. So in the fall season, again, the tomato beds, we collected samples for analyzing nitrate and ammonia and organic matter. So these are data from nitrate and ammoniacal nitrogen, which is in the top 30 centimeters of the soil, which is kind of plant available for the tomato. So you can see here in both, this is data from 1 year, but the same trend we found in the next year for nitrate as well as ammonium. There is a numerical increase in that nutrients in the bed with cover crop and compost trial. When we looked at the organic matter, the higher organic matter was found, numerically higher organic matter was found in the compost only treatments, which makes sense. Again, compost is adding organic matter. So this is also a baby step towards integrating these practices. Yes, it might help. It can help retaining some of the nutrients for your crop production season. So that's something I want to show you. Then we want to know if these observations were translated into the yield. So we took the yield data in 2 years, and you can see here in the first trial, the cover crop and compost treatment, we had a significantly higher yield. And the next year, actually, the second trial, we did not have a significant increase, but there was a numerical increase in the yield as well. And if you look, another interesting point I want to bring to the table is the compost only. Again, you have a lesser yield in both trials because it might have been boosting the weeds and reducing the yield. So weeds, when you have more weeds, it's going to compete with the crop and impact the productivity, right? So that was the whole crux of this study. So just to summarize it up, or the take-home messages include composting. When you combine composting with cover cropping, yes, it can boost the cover crop in the summer fallow. And also, we also noticed a reduction in the weed pressure in the subsequent season in the fall with some cover crop alone, or when we mixed it with composting. But compost only application, it did not do a great job. Especially, it boosted the weeds and reduced the yield. And slight increase in the nitrogen was observed in the cover crop and composting, as well as, and they also help improve can potentially help improve the productivity, as we observed from the yield as well. So that's all the highlights I want to share with you. So again, Didrot is a student who collected all this data. So thank you, Didrot. And I want to thank all these folks, especially my collaborators and my team. And they do all the job. I'm just presenting here. So thank you very much. We have a weed garden in the back of our station if you want to take a look. So I will stick around if you have any questions. Thank you.
Thank you, Ramadas. Any questions as we switch here? No? All right. So our next presenter is Nathan Boyd from Gulf Coast Research and Education Center. He actually ended up having Congressman is visiting their center today, our second congressional leaders. So he's going to be giving it remotely, his presentation. So next up is Nathan Boyd, and he's going to be talking about his work in Fumigants. Nathan, go ahead.
It's a pleasure to be here today, everyone. I appreciate you taking the time, and I am sorry I could not be there in person. I would like to have been. I'm going to spend a few minutes talking about weed control with fumigants. And as most of you know, we have a very select number of fumigants that are typically used in vegetables, which consists mostly of different ratios of chloropicrin and 1,3-dichloropropene, and as well as metam potassium or metam sodium. There are a few others here and there, but those are the most common fumigants used, and pichlor 60 in vegetables is by far the most common of all of them. So today I'm going to talk about two scenarios: weeds that emerge in the planting holes, like you see here, where you see this weed growing along with tomatoes, and of course as well our favorite critter, the nutsedge that can puncture the plastic mulch. And with that, I do want to just first say just a real quick couple of comments about plastics. And in the picture you see here on the left is no mulch. These are all together kind of randomized in a field, and the use of plastic, even if it doesn't really matter what type of plastic you use. This is an LDPE, this particular picture. The use of plastic is one of its major benefits is the control of broadleaf and grass weeds. And specifically, we know that nutsedge can puncture nutsedge, but we often forget that it still can reduce the density of what's actually growing there. In some cases, that's not true, but often it does. And so what it's doing is basically it's just functioning as a barrier. And then you have TIF. TIF does a few things. Number one, it holds the gas in the bed longer. If you hold the gas in the bed longer, you get better weed control. So generally speaking, with TIF, you're still going to see better weed control even in the planting holes of broadleaf weeds and grasses than you would with a VIF. Most people think of the use of TIF for limiting nutsedge, but it actually, our studies show it does reduce overall broadleaf and grass control as well. And then TIF also inhibits nutsedge penetration. The mechanism I'm not entirely sure of. We've tried a few different trials to try to figure that out. The one thing we do know for sure, we just finished some trials last year, is that plastic thickness has very little effect on nutsedge penetration. And we were using a whole range of thicknesses. Of course, I mean, you could at some point get thick enough where it would, but with the types of plastics that are on the market for use in this type of situation, we don't think that, we didn't see any difference in the trials we ran on nutsedge penetrating through the mulch. And it's important to note that TIF, using that plastic, does reduce nutsedge density even in the absence of fumigants. So it's not just that it holds the fumigants in the bed longer, it actually does inhibit it in some way. A few years ago, we did a lot of work looking at fumigant mixes and their effects on weed control, and we did a bunch of studies on commercial fields around the state, looking at some general patterns. And here is just a couple of pictures that kind of demonstrate a consistent pattern that we see. On the left is a picture of a non-fumigated plot. This is in tomatoes, and you can see there's a lot of nutsedge here. In the center, you can see 50 pounds of pick, and in fact, the nutsedge looks worse. And this is common trend. Really low rates of pick can actually stimulate sprouting of the tubers and can make more nutsedge present. Picture on the right, this is 200 pounds of pick. So once you get that rate up to a reasonable level, there's still nutsedge in that bed that you can see, but pick does suppress nutsedge on its own if you have a high enough rate. Now, two pictures on the left and the center stayed the same. Here, I've just put, changed the picture on the right, and this is 50 pounds of pick combined with 1,3-D, and immediately you see a distinct difference. There is no nutsedge on this bed, and if you look further back in the picture, you can see a perfect line where we shut it off. And the only point I'm trying to make with these pictures is to demonstrate the fact that combinations of fumigants, pick and 1,3-D, for weed control always work better than relying on one component of its own. So just using pick or just using telone, you're not going to get the same level of weed control as you get when you combine them. So nutsedge management generally improves when chloropicrin is added to the combination, and this is true across the whole range of fumigants. When you add some chloropicrin, we think it helps stimulate sprouting, which then the other fumigant improves its efficacy and kills more. And as I mentioned, a combination is almost always better. All right, we also looked at some work on Picklor 60 rates, and of course this has been done for a long time, and it really wasn't the rate data that turned out to be interesting. It was that we were able to prove something that we've long thought to be true, but was finally able to have the data to prove it. This graph, if you look across the bottom, this is Picklor 60 in pounds per acre. So all the way from nothing to 150 pounds, and then we went all the way up to 500 pounds per acre of Picklor 60, which is, that's a very, very high rate. And then on this graph is the number of nutsedge. This is number per square meter of nutsedge in this field. And then the two lines. This line right here is a fall fumigation. So at this particular site, this is in Central Florida, this was done in July. And then if you look down here, this fumigation was done in January. And first of all, you have more nutsedge in July than you did in January, but also the ability to control that nutsedge. So what we found is application of Pickler 60 in cooler temperatures that you would see in January works a lot better than if you apply that fumigant during the really hot temperatures when that fumigant's moving out of the bed a lot faster. This is looking at, we buried yellow nutsedge tubers in the bed and looked at, after the fumigant had been in there, we retrieved them and then put them in conditions where they would sprout and looked at the number of sprouting. So basically, this is, rather than just looking at nutsedge control, we're looking at how many of the actual tubers were killed. And you see the exact same pattern. Basically, even 150 pounds per acre of Pickler 60 in January, everything on up, there's complete, all the tubers were killed that we had put in there. But if you look at fumigation in July, there's a lot of variability. You see these wide error bars, that means there's a lot of variability from plot to plot. And it took up to 300 pounds per acre before you were getting really good control of those tubers. And that's kind of, again, the same difference. You might get the same level of control, but you need the higher rate at the higher temperatures when you're fumigating. Saw a very similar pattern with, this is tomato yield. And you can see that with this bar that you're getting, this is in January, you're getting a higher yield initially. It's also important to note at the really high rates, though, you start to cause more damage on the crop and your yield starts to drop down. Of course, this is, very, very high rates. Now, if you have, just kind of want to remind you, this picture that I showed you early on where we have the non-treated, the pick on its own 1,3-D, you can see the plastic is black. That's because we're fumigating here in January. If you take, I'm just showing you two pictures on the right here, this is a non-fumigated, plastic's white. This is because this is when we're fumigating in July. White, and then you look at Pickler 60, which is that combination of 1,3-D and pick, and it's just full of nutsedge. And so early on, when people described how fumigants work, they always said, well, fumigants have a variable response. Nutsedge control is variable. Well, it might be variable, but it's not random. It's very dependent on the soil, moisture, and temperature. So the level of control you achieve in experiments is going to, and in your field, is going to depend on when you actually fumigate. And a lot of that, part of that has to just do with this. Sprouted tubers are much more susceptible to fumigants than these ones that are not. And that's probably just because you have more surface area that can absorb the fumigant. But if the fumigant's held in the bed longer or stays in the bed longer, which you can achieve with TIF, but also you can achieve that in cooler temperatures when the fumigants don't move as fast, then it's more likely that tubers are going to sprout while the fumigant's still there, and therefore more likely you're going to get better control. We did a lot of studies looking at ratios on, as I mentioned earlier, on commercial farms. So these are really large plots. And I'm not going to go through this whole table. I just want to show you that we used all these different fumigants at the same sites. So from Telone 2, C17, C35, Pickler 60, Pick 80, and Tri-Pick 100, with the idea that we're going from no chloropicrin and all 1,3-D all the way up through all the commercially available ratios to 100% chloropicrin. And then in the strawberry trial, we had the presence or absence of K-PAM as well. And I want to show you a couple of pictures because this is really interesting, and this trend is pretty consistent. So the top picture on the left-hand side, this is a non-treated control without K-PAM. And you can see these plants are really small and stunted. This is strawberries. When you add K-PAM, there's still some weeds in there. You don't get great weed control. K-PAM on its own doesn't do a great job in nutsedge, but the plants are a lot healthier, a lot bigger. And then if you put Telone 2 without K-PAM and Telone 2 with K-PAM, there's actually more weeds in the bottom corner. Not really seeing any benefit of adding K-PAM in terms of weed control when it's added with Telone 2. But if you look at C15, so it's just the pictures on the right that are changing, really bad nutsedge, adding the K-PAM here maybe helped a little bit, but not a lot. But this is where you start to see the difference. When you have C35, so a combination that's a little bit more closer to equal ratios of PIC and 1,3-D. Actually adding KPAM in that situation gives you added benefit, is where you really see the KPAM have an additional benefit in terms of weed control, which you can see here, there's basically no nutsedge in this bed. And that trend holds true with PIC 60. You can see how bad the nutsedge is here with no KPAM. Then you add the KPAM, the plots look really good with PIC 60. And all the way on up, here we don't have a lot of nutsedge in either plot, but if you get to TriPic 100, TriPic 100 plus KPAM has worked better than Telone 2 plus KPAM, but still TriPic 100 did not work as well as having a combination of PIC and 1,3-D. And this is crossed, if you look across all of the ratios of PIC and 1,3-D, the nutsedge density with KPAM is significantly lower than without, as you would expect. When it came to the number of weeds per plot, though, broadleaf and grasses, a lot of variability, but no consistent pattern really across these different fumigant ratios. So just in summary of this particular set of work is that supplemental KPAM improved weed control across most fumigants. Yields also tended to be slightly higher. And this is kind of a pattern we've seen. This work is in strawberries, but we've seen it in tomatoes. We've seen it in peppers. We've seen it in cucurbits. That Florida three-way, which is basically Picklor 60 injected into the bed and then KPAM placed with closer to the surface of the bed, consistently gives the best weed control and consistently gives the best crop yield. We did a long-term fumigation trial. When I say long-term, this is 4 years, same fumigants on the same plots every year. And we were looking at a non-treated control, Telone C35, Picklor 60, chlorpicrin on its own, KPAM on its own, and then Picklor 60 plus KPAM, which is the Florida three-way, and you can see the rates here in pounds per acre of what we used. Basically at Telone we're at 225, Picklor 60 we went 275, TriPic we went 300 pounds, KPAM is at 40, and then Picklor 60 plus KPAM was 275 plus 40. The 40 of course is gallons per acre, not pounds per acre. And these are kind of rates we would recommend. This is all done under TIF. And just going to show you some pictures that are representative of the data we analyze. As you would suspect, this is done in a field with lots of purple nutsedge. So if you didn't fumigate, you got a lot of nutsedge happening. Picklor 60 worked really well. Notice this is fumigated in January, so that's why. KPAM on its own did pretty decent compared to the non-treated, but still a little bit more nutsedge than Picklor 60. And then the Florida three-way is completely clean, no weeds at all in this particular scenario. And these pictures were taken right after transplant. If you look at harvest, the non-fumigated, the plants were, I mean, we had bacterial wilt and the crop would have been nothing, which is a good illustration of why we fumigate in Florida. Picklor 60, you see a nice, really healthy plants looking really good, really robust. KPAM, you can see it did not hold. Plants look good, but a lot of weeds in those plots. And the Florida three-way, by far the thickest, healthiest plants, highest yield, and the cleanest plots in terms of weed control. So to summarize this, the fumigate combinations maximize weed control. Most consistent weed control is in cooler temperatures. Florida three-way is the most consistent fumigant in terms of nutsedge control, yield, and we did one economic analysis and it was also gave the highest return. And the Florida three-way also provides the most consistent broadleaf and grass control. And I want to just end this with one slide on cover crops. I know Ramadas had already, I got to listen in, he talked a little bit about that. And I just want to point out that we did this trial where we did the same fumigants for multiple years. We also did the presence or absence of a cover crop. And our results kind of shocked us, but it was this consistent across years. Every year was the same, that actually the presence of a cover crop had a bigger impact on the weeds than which fumigant we used. And that just highlights the real importance and value of cover cropping. So I want to thank Trias and AMVAC and also the FDACs who have supported this and an amazing team I have here at GCREC. And I appreciate you taking the time to listen.
All right, and to wrap up our morning together, our programs today, we're going to get to hear from our sponsor.
Okay, good morning. Can you all hear me? So I just want to say, first of all, to start us off, a big thank you to Craig and the University of Florida for allowing us to come in and sponsor today's event. We're so excited to be here and proud to be collaborating with the University of Florida along with the growers down here in the Southwest Florida region. I'm going to keep it pretty brief here so we can get into kind of the technical aspects, but I'll give you a quick overview of ViaFlex, our company, and what we do. First, can I get a show of hands? I just want to see who's here, who's using row mulch plastic in their fields. And maybe there's not many of you here today. In this region overall, could you tell me, is it like 60%, 70%, would you say, are using some kind of a film, whether it's TIF or non-TIF? Depends on the crop. Okay. Sounds good. All right, so I'll just give a quick overview here. So the mission here at ViaFlex, right, basically competing by developing innovative solutions and exceeding our customer expectations for quality and service. I'd say that's the main one, right? You know, we're here to, at the end of the day, provide the growers and those that support the growers, like the University of Florida, with an innovative product that's going to give you a higher yield, better crops. Right. And so the films, and I liked how we followed Nathan's presentation where he talked about how, you know, they're using the different fumigants, the cover crops, but then also how important the films are, which we'll demonstrate today in our presentation. You can go to the next slide. So we were formerly with Raven Engineered Films. Anybody in here familiar with Raven or had heard of Raven? A few of you maybe. Yeah. Yep. So we used to be with them. And then back in 2022, we were publicly traded. So ViaFlex now is a private standalone company, which is exciting for us. Even though our name has changed, right, we still have over 60 plus years of experience to draw on when it comes to engineered films. So basically what that means, we are developing and manufacturing thinner, lighter, and stronger products, in this case, the row mulch that's here today, to help solve application and product challenges. Right now here, moving into the Southeast region. So we have a big presence in Southern California, mainly. Historically, we've been working with growers out there. But really looking ViaFlex, now that we're a standalone company, we really want to look at the market and say, where else can we have an impact? In the Southeast region, you know, is huge, from Texas to the Carolinas, Florida. So that's why we're here today. And then there's another one. So before I hand it off to Daniel, or to Derek, excuse me, I'll go ahead and I'm going to pass around as he speaks. This is a non-TIF 1 mil film that we have. So I'll just pass this around as he talks about kind of the technical details here. And then I'll also pass around a flyer that kind of talks about the difference between row mulch, the blown versus the cast. So this film here that we have today is a blown film. I know most of you here are used to using either cast or embossed, but we really feel that a blown film has a place in the market as well. So I'll go ahead and hand it over to you, Derek.
So my name's Derek Weaver. I'm part of the R&D and product development team. Thanks, Tess. So my name's Derek Hoover, part of the R&D and product development team at ViaFlex. Like Tess mentioned, used to be part of Raven Industries, which is now owned by Case New Holland. So very familiar with, I'm sure people that raise their hands probably know about the Precision A component. Raven Engineered Films broke off earlier this year into ViaFlex. And we've been around for, 60 years, like Tess mentioned, and have a strong presence out west in roll mulch films, primarily focused on the TIF or the barrier film. So Nathan kind of showed the value adds that you can get from using TIF products with the amount of fumigant that you're applying, as well as being able to retain that fumigant into the soil to help mitigate nutsedge and other weeds from puncturing through the plastic and interfering with your overall yields. So there's a couple of different manufacturing methods, and Bioflex actually has equipment for both of these. What's commonly used out in the marketplace today on the roll mulch, whether it's TIF films or the standard low density polyethylene films, a lot of the films are coming through a cast extrusion process, which would be that picture in the top left up here. And I just want to take a couple minutes to explain that process and a blown film process and some of the pros and cons or advantages between the two. So starting with the cast process, basically you will have a heated screw here that's going to basically melt down your polymer, go through the barrel, and it's going to come out a hot dye. It's going to be very hot at that point. It's going to be molten. And right away, it's going to be cooled down on a chilled roller within seconds of it coming out of that dye. And then it's going to be pulled through a series of winders back to the final winder where it's going to be rolled up. And what that does through that process is creates at the molecular level orientation in the direction that material is being pulled. So in this case, it would be in the machine direction or the length of the roll as you're laying it out in the field. So that means that you're going to have your molecules aligned in that direction, and you're going to have good properties along that direction in the machine direction. Now, you also have a blown film extrusion process, which is similar, but instead of having the molten polymer come out of a die and go downward and get cooled rapidly right away, we're actually blowing upwards or vertically. So you have a circular die that you're still going to have that molten polymer come out of. But you see here, it also goes out at an angle. So you're going to blow it up essentially to whatever circumference. So if it's 60-inch lay flat, you're going to extend that material out in that direction. At the same time, you have a nip that's up top that's going to pull that material vertical. So what that does is give you a more randomization of that molecular alignment. So you're not only going to get the alignment in the machine direction, you're also going to get some diagonal alignment of those molecules as well. So overall, you're going to get more balanced physical properties of the film in both directions. So what does that mean for a grower or somebody that's going to be laying down the roll mulch into the field? Well, a few different things. If you have a lot of tension when you're deploying that roll mulch film, and say you get a puncture through it during that process, a lot of times once you get that puncture when it's under tension, you'll see a zipper and just go right through it. And that's really common with the cast roll mulch films. So what they do to try to accommodate for that or try to improve that is they'll put a micro embossment into it to help kind of impede that zippering. Now with a blown film, you get not only the strength in the machine direction, you also do in the transverse direction. So what that's going to give you is superior puncture strength. If you do get a puncture, it's not going to tear as easily because you're going to have strength in the other direction as well to help kind of truncate the overall length of that tear. And it also gives you some down gauging options because it's going to have higher physical performance. Maybe you're used to using a 1.5 mil or a 2 mil roll mulch film. You would be able to achieve similar performance at something that's about 0.8 to 1 mil with a blown film option. And it also negates the need for that micro embossment. So another advantage with having that physical strength in the 90-degree direction or the transverse to the bed is you can stretch it over that bed without tearing it because of the tensile and tear properties it has with that. So it's going to lay flatter. It's going to have a tighter bed formation. And again, we have both manufacturing processes, but we really like to tote the blown film because of those physical properties that you can gain from using material produced through that process. So that's really I know we're right in between you and lunch, but we really wanted to just kind of introduce Bioflex, formerly Raven, and kind of highlight some of our manufacturing capabilities. And we definitely appreciate the time to get in front of all of you. And if you have any specific questions, feel free to ask now, or we'll stick around as well and come reach out to us. So we have both standard options, and we have TIF, and we have VIF. So it just depends on what the needs are for the application. So we offer both. The standard film is going to be a polyethylene. Generally, it's going to be a linear low density polyethylene. Sometimes it's a mix, depending on, again, what the need is of a low density polyethylene as well as a medium or high density. So our capabilities, we can actually have 9 different layers built into the film structure. So we can have layers in the core that are going to provide the TIF properties for the fumigation. We can have layers on the external surface that might be more need more toughness or abrasion resistance. So we can fine tailor each layer of the overall structure to meet a different need or solve a different problem. Any other questions? If not, again, we'll be sticking around. So if there's any questions that come up, please do reach out.
All right. Tess, Derek, thanks again. Please thank them again and thank them throughout the lunch. They made this possible for us to meet today, and they provided the lunch for us as well. So please make sure you thank them. So with that, we're going to conclude our meeting.